All right, welcome. Today, we're pulling off a heist, a microscopic one. Our target, a cell's most valuable secret, its complete DNA blueprint. So let's gear up and crack this code.
I mean, really think about this for a second. A single bacterium, it's like a tiny fortified vault. And locked deep inside is the very code that makes it tick. So getting it out, it's not just some simple procedure. It's this fascinating puzzle that takes precision, some clever chemistry, and yeah, a little bit of brute force.
Every living thing, from you and me to the tiniest bacterium, runs on a set of instructions. And that instruction manual, that's its DNA. But here's the thing, you can't read a book while it's still being printed and bound inside a factory, right? To study it, scientists first have to pull that manual out, cleanly separating it from all the other humming machinery inside the cell. So that's our goal for this explainer. Simple enough on paper. We're starting with a single bacterial colony, and by the end, we're going to be holding its pure, isolated genetic code. Let's get started with phase 1 of the operation. So our first challenge is just getting past the cell's tough outer layers. A bacterium has a cell wall and a cell membrane, and they're both protecting its precious cargo. To get to the DNA, we have to breach those defenses. This is the break-in. Our break-in plan is basically a three-pronged attack. First, we get the cells into a liquid suspension. Then, we hit them with a chemical cocktail that's designed to just tear down the walls. And finally, we give it some time, let it incubate so those chemicals can really do their work. Our first tool is something called TE buffer. Now, a buffer is really just a solution that keeps conditions stable. So you can think of this as picking the first lock. It protects our DNA from getting damaged while it starts to weaken the cell's outer wall, getting it ready for the main assault. Okay, next up in our toolkit is SDS. You can think of this as our chemical battering ram. SDS is, well, it's basically a really, really strong soap. And you know how soap just dissolves grease on a dirty pan? That's exactly what SDS does to the cell's fatty membrane. It just dissolves it, causing the whole thing to fall apart. Boom, the vault door is officially open. But even with the door open, the DNA is still surrounded by guards, in this case, proteins. That's where proteinase K comes in. It's our molecular wrecking ball. It's an enzyme that acts like tiny scissors, chopping up those protein guards into harmless little pieces. And we incubate it at 37 degrees Celsius, that's about body temperature, because that's the perfect warm environment for this enzyme to do its job. So we've broken in, and we've neutralized the guards. But now, the vault is a total mess. It's full of busted up cell walls, dissolved fats, and all those chopped up proteins. Our prize, the DNA, is mixed in with all of that junk. So, it's time to call in the cleanup crew. The cleanup involves another chemical cocktail. First, we add some special salts that basically make all the cellular junk, all that debris and protein, clump together. Then we add a mix of chloroform and isoamyl alcohol, which is going to act like a separator, pulling all those unwanted clumps out of the watery solution where our DNA is hiding. So how do we actually separate the junk from the treasure? We use one of the most important tools in any biology lab, a centrifuge. We're going to spin our tube at an incredibly high speed to force everything to separate based on how dense it is. Okay, so spinning this thing at 12,000 revolutions per minute forces the whole mixture into 3 distinct layers. On the bottom, you get this heavy organic layer with all the junk we want to get rid of. In the middle, there's a messy interface of cell debris that you definitely have to avoid. And on top is a clear, watery layer. That is our prize. It contains the pure DNA. We very carefully pipette it out, and we actually repeat the cleanup skin a second time, just to make sure our getaway is perfectly clean. All right, this is the moment we've all been waiting for. We have this clean liquid, and we know it contains our DNA, but we can't see it. The DNA strands are just dissolved in the solution, making them completely invisible. So it's time for the final, and honestly, the most dramatic step, the capture. So far, we've been kind of working on faith, right? Following the steps, trusting that the DNA is in there somewhere in that clear liquid. But now we get to see the fruits of our labor. We're about to perform a little bit of chemical magic. And this is it. We add isopropanol, you probably know it as rubbing alcohol, to our solution. Now here's the cool part.
DNA absolutely cannot stay dissolved in alcohol, so almost instantly, these ghostly, stringy white threads just appear out of the clear liquid. That's it. That's the DNA. It's clumping together, becoming visible to the naked eye for the very first time. The genetic code is literally materializing right there in the tube.
We are in the home stretch now. We spin the tube one last time to collect all that stringy DNA into a tiny white speck at the bottom, which we call a pellet. Then we give it a final rinse and let it air dry. It's kind of like washing and drying our captured treasure to make sure it's absolutely pristine. And that final rinse uses 70% ethanol. Its only job is to wash away any leftover salts that might be clinging on, making sure what we have is nothing but pure, clean DNA. And with that, the heist is complete. We have successfully broken into a cell, cleared out all the junk, and captured its genetic code. But that raises the most important question of all. Now what?
After all that work, we take our tiny, dry pellet of DNA and we redissolve it in a minuscule amount of stable buffer, just 50 to 100 microliters. To put that into perspective for you, a single teardrop is about 50 microliters.
And that's what's so incredible. Inside that tiny, clear drop of liquid is the entire instruction manual for a living organism. Millions of letters of genetic code, perfectly preserved and ready to be read. You see, this whole process, it's the foundation of modern biology. By extracting DNA, we can do amazing things. Identify new species, diagnose diseases, engineer life-saving medicines, even solve crimes. We've captured the code, so the only question left is, what secrets will we ask it to reveal?